Hi, I'm Nikki Bradley. I'm the module leader for the Physiological Principles module. I'm also the Deputy Course Director for the ACP course. Welcome to this three-part preparation for the physiology module. Please watch all of the videos in sequence, make any notes that you need to make, and please feel free to stop the video whenever you need to make notes or you need to think about something that I've said. Meanwhile, enjoy.
So learning outcomes. At the end of this part 1 of 3, you should be able to define the term atom, elements, molecules and compounds, and describe the structure of an atom. State the most important biological elements and suggest how atomic weight and atomic numbers are derived. Discuss the significance of the outer shell electrons, the valence shell electrons, are to reactivity of elements, atom binding to atom. And discuss ways in which atoms bind to form molecules and compounds. Atoms. All matter is composed of substances called elements. And matter is basically anything that occupies space and has a mass. Elements are unique substances that cannot be broken down into simpler substances by ordinary chemical methods. And new elements are still being discovered. Each element is composed of atoms. Atoms consist of subatomic particles called protons, neutrons, and electrons. The atom has a central nucleus, which has positively charged protons and neutral neutrons. The atomic number refers to the number of protons within the nucleus. So, for example, oxygen has an atomic number of 8, so it has 8 protons. Hydrogen only has 1 proton, so its atomic number is 1. Carbon's atomic number is 6, therefore it has 6 protons. Orbiting around the nucleus, there are the negatively charged electrons. 24 elements essential for the human body are the ones here. So at the top, you can see on the table, you have oxygen. And the table descends in order of their significance, really. So look at the first 3 at the top. These are the 3 most prevalent elements of the body. So we have oxygen, carbon, and hydrogen. So remember, these elements are made of atoms, so they can be combined to form molecules. Thinking about the first one and the third one, hydrogen and oxygen can be combined to form, yes, water, H2O. 2 hydrogens, 1 oxygen makes water. In the atom, the number of protons, neutrons, and electrons are usually the same. Remember, protons and neutrons are in the nucleus, and electrons orbit the nucleus. And remember that the number of protons give the element its atomic number. The exceptions are isotopes. In isotopes, there is the same number of protons in the nucleus as the atoms of that element, but they have a different number of neutrons. The electrons stay the same. So in the isotope, the number of protons stays the same, so the atomic number doesn't change. So a good example is carbon. Carbon has 6 protons, so its atomic number is 6, and usually has 6 neutrons. Carbon-14, as you know, used for dating things, has 8 neutrons. So it's 6 plus 8 gives it the number 14. The 6 being the protons, the 8 being the neutrons. Ions, here the variation is in the number of electrons. So in electrically neutral atoms, the number of protons in the nucleus, as we said, is the same as the number of orbiting electrons. But if you have a variation in the number of electrons, it will create an ion of that element. I-O-N, not I-R-O-N. The protons make the nucleus positively charged, but this is counteracted by the negatively charged orbiting electrons. Remember, we said that protons are positively charged and electrons negatively charged. Neutrons, of course, being neutral. Because opposite charges attract, the electrons are bound to the positively charged nucleus. Remember what happens with magnets. If you try and put two magnets together using the ends of the same charge, they repel. So if you put a positive pole to a positive pole, they'll push apart. If you put the negatively charged end to the positively charged end of another one, they'll snap together and grip. This is because opposite electrical charges attract. And this is what happens to keep the electrons bound to the ion. They're attracted to the positive nucleus. In an atom that has an equal number of protons and electrons, it's electrically neutral because they counteract each other, the charges. However, a cation has lost one or more electrons. So therefore, because it has more protons than electrons, it becomes positively charged. Good examples here are sodium, Na, which will become Na plus, a cation of Na, when it loses an electron. Notice here we're using the chemical symbol and a small plus to denote it. And this is because it's more precise than just writing sodium, because if we write sodium, we have to write the word cation. So it's much easier and more accurate to use the chemical symbol and the little plus symbol. Calcium becomes Ca++ or Ca2+ when it loses 2 electrons. An anion has gained an electron, so it becomes more negative, so it becomes negatively charged. Good example of this is chlorine. Chlorine becomes Cl- when it gains an electron. Of course, it can't lose a proton, but it can gain an electron to make it more negative. And again, notice the symbol. We've got Cl for chlorine and a little minus to indicate that it's an anion.
Okay, so let's have a look at our atom. So as we said, we have a central nucleus. Within that nucleus, we have an equal number of neutrons, which are, as the name would suggest, neutrally charged. So in other words, they don't carry a positive or a negative charge, they're just neutral. Also inside the nucleus, we have positively charged protons, an equal number in a normal atom to match the neutrons. So, okay, where are the electrons? The electrons orbit the nucleus in a shell, or in multiple shells. So we might have one shell orbiting around. We've got 2 electrons. We know it's 2 because the innermost shell can only take 2 electrons. So let's have 2 electrons. There we go, 2 electrons. Because we have an equal number of protons, neutrons, and electrons in a normal atom, we now need another shell, because the maximum the innermost shell can take is 2. So let's put another shell in. Okay, equal number, we're going to have 1 electron. Now we have 3 negatively charged electrons and 3 positively charged protons, and 3 neutrons that just sit there doing nothing. These 2 are counteracting each other. So our atom at this point is neutral. We have an equal number of electrons and protons. However, okay, imagine we add an electron. So let's put an electron in. That electron is negative. Therefore, we now have 3 protons that are positive. We have 4 electrons that are negative, more negative. Therefore, we have, yes, we have an anion. It's an ion that is negative. Okay, conversely, let's take 1 electron away. This gives us back to a neutral atom. Let's take another electron away. We now have 2 electrons, 3 protons. Protons are positive, electrons are negative. Therefore, this is now going to be less negative or more positive. In other words, we have a cation. Both of these are ions. Cations are positive, anions are negative.
Hydrogen only has 1 electron. It also only has 1 proton. If you remove the electron, it becomes a cation of hydrogen. It becomes H+. H+ is often called a proton because that's all it has. It's lost its 1 electron and all it's got left is 1 proton. And you've probably heard of proton pumps. Proton pumps are cellular transport mechanisms that pump H+ across membranes. Electrons travel around the nucleus in orbits or shells. Each shell has a limit as to how many electrons it can contain. The closest to the nucleus can only contain 2 electrons. The next shell can hold 8, and then the next one 18, and so on. So as the shells fill up, another shell forms. The outer shell is called the valence shell, and here electrons like to be in pairs. If the valence shell is full, the element is considered to be inert because it can't form bonds with other atoms in this state. And most inert elements are gases. This forming of bonds is what happens to create molecules. So atoms will bind to another atom to form a molecule. Molecules are formed then by atoms binding together. Covalent bonds between atoms involve the sharing of valence shell electrons between 2 atoms. This binds the atoms together to form molecules. So the molecule that we've got on the right, we've got 2 oxygens and 1 carbon. So we've got, yes, carbon dioxide. Molecules can have a double covalent bond with 2 shared pairs of electrons, or even a triple where there are 3 shared electrons. Have a look at the carbon dioxide above and also the oxygen molecule on the left. What type of bonds do they have? Yes, they're both double covalent bonds. Note that oxygen is a molecule because it has 2 oxygen atoms bound together, hence O2. Ionic bonds. These occur where the electrons are not so much shared, but they're actually transferred. This makes the 2 atoms of opposite charge, so they attract each other, and this creates the ionic bond. For example, sodium chloride forms crystals through ionic bonding, and sodium chloride, of course, is, yes, salt. Na only has 1 valence shell electron. It donates this to chlorine, and this forms this ionic bond that creates the NaCl, that binds Na to Cl. Look at the diagram on the right. Sodium, Na, in purple, gives its 1 outermost valence shell electron to chlorine, Cl. But it's now attracted to it and forms an ionic bond to it because Na has become more positive. It has more protons than electrons, so it becomes a cation. And chlorine has become more negative. It has more electrons than protons, so it's an anion. Remember, opposite charges attract, like the plus and minus ends of magnets. But in solution, water breaks the ionic bonds of NaCl. Think what happens when you put salt into water. It dissolves. Cl keeps its electron, making it an anion, so it becomes Cl-. Na has lost its electron to Cl. This makes it a cation, so it becomes Na+. Na+ therefore becomes a separate cation, and Cl- becomes a separate anion, which breaks down those crystals of NaCl. Na+ and Cl- are 2 of the most important ions in the body. Water is what's known as a polar molecule. Atoms within any molecule may have fewer electrons in their shells than other atoms within that same molecule. Fewer electrons makes them slightly more positive than others that have more electrons. So in terms of water, hydrogen only has 1 electron. Its atomic number is 1. Of course, the 1 refers to the protons, but as long as we're not talking about an ion, the electron number is the same as the proton number. Yes, so because it only has 1 electron, it's more positive than oxygen. Oxygen has 8 electrons. Its atomic number is 8. It is more negative, therefore, than hydrogen because it has 7 more electrons than hydrogen does. In terms of the water molecule itself, 2 hydrogen atoms bind to 1 oxygen atom to form a water molecule. i.e. H2O, hence the 2 after H. Because hydrogen only has one electron, it wants to share one of oxygen's electrons to give it a pair of electrons in its only shell. Hence, it binds to oxygen. This is actually covalent bonding because water shares an electron with each hydrogen atom. The south pole of a magnet is negative, the north pole is positive. So comparing that to our water molecule, remember our oxygen is more negative because it has more electrons, whereas our hydrogens have fewer electrons, they're more positive. So the water molecule acts like a little magnet, the oxygen side being negative compared to the hydrogen side. So water is very like a little magnet. One pole, the hydrogen side, is more positive than the other. Hence the term, it's a polar molecule. Surface tension is caused by hydrogen bonds. This is where you have molecules of water being attracted to each other. So the more positive hydrogen side of one molecule is attracted to the negative pole or side of another water molecule. And this is what gives water surface tension. The positive hydrogen side of the molecule is attracted to the negative oxygen side of adjacent molecules. Remember that opposite charges attract, and this is a weak form of bonding called a hydrogen bond. Water will support light objects and form droplets because of this surface tension. Water molecules also form hydration shells around ions. It does this because the negative pole of water, i.e. oxygen, is attracted to cations because they're of opposite charge. Also, the positive pole of water, hydrogen, is attracted to anions because, again, they are of opposite charge. This is why water can break that ionic bond that forms NaCl. It also applies to potassium chloride, KCl, which becomes K plus and Cl minus in solution in water. The outer charge of this hydration shell around an ion remains the same as the ion because of the way opposite charges attract. So our negative oxygen is attracted to our positive sodium, but of course the other side of it is positive, so the outside of the hydration shell will be positive, like the sodium. Hydration shells, however, have to be removed before ions can get through ion channels in the plasma membrane of cells.
Okay, so let's have a look at how water interacts with sodium chloride to give us sodium and chlorine. We know water is a polar molecule. So we have oxygen, we have two hydrogens, hence H2O. Yeah? Two hydrogens. Hydrogens have fewer electrons. In fact, they only have one each. Oxygen has more electrons, therefore, of course, it is more negative than hydrogens. So we have a polar molecule. This side of the molecule is positive compared to this side, which is negative. And that's what happens, well, that's what happens with surface tension, remember. So oxygen, which is negative, opposite charge is attracted to hydrogen, so all the little water molecules stick together. In terms of NaCl, what we have is we have Na and Cl. We'll make them different colours so it's nice and clear. Cl, Cl. There we go, nice little electrons. And of course, we're sharing an electron. Or in actual fact, really, it's an ionic bond, so sodium has donated the electron to chlorine, but it's hanging on to it. Sodium and chlorine gives us NaCl. However, when you put that into solution, most of it, water will get in the way, basically. Water is attracted to the two molecules. So we end up with Na, who has donated his electron. So he is now Na plus. He has donated his electron. He is positive. He's a cation. And we have Cl, which has gained the electron, therefore it has more electrons, so it is minus. It is negative. So, okay, in terms of our water, what do you think is going to be attracted to what? We've said opposite charges attract. So, oxygen, which is negative, is going to be attracted to our sodium. So we're going to end up with oxygen and two hydrogens sticking out like that. Oxygen, two hydrogens, oxygen, two hydrogens, oxygen, two hydrogens, and so on and so forth. This is giving us this hydration shell. Water has come between the two. Okay, in terms of chlorine, what about chlorine? Chlorine is negative. What's chlorine going to be attracted to? It's going to be attracted to the hydrogens. So in this case, we're going to have two hydrogens and the oxygen. Two hydrogens and the oxygen. In this case, we've got the negative pole attracted. In this case, we've got the positive pole attracted to chlorine. Consequently, our NaCl has become Na plus and Cl minus. And these are two of the most important ions in terms of the body.
Osmosis is about the movement of water, not of solutes. Solutes of a higher concentration, however, attract the water, and solutes basically can be all chemicals. So solutes can be ions, molecules, even macromolecules like glucose. The concentration of solutes between two different compartments is determined by water following an osmotic gradient. Water moves from an area of low osmolarity, so fewer solutes, to an area of higher osmolarity, where there are more solutes, to try and equalize the osmolarity of the two compartments. And this is a bit like adding water to concentrated squash to dilute it. In the body, the compartments are the intracellular, so within cells, and the extracellular. And this includes the blood and also the interstitial fluid around the cells. In terms of these compartments, they are usually osmotic equilibrium. So this is known as isotonicity. There is no osmotic gradient. The amount of solutes on either side between the intracellular and the extracellular is exactly the same, so there's a net movement of water. Water doesn't move. In hypertonicity, cells will shrink, and this is where there is a higher number of solutes outside the cell, so water moves out of the cells to try and dilute it, and the cells shrink. Hypotonicity is where cells swell, so there is a higher amount of solute inside the cell, so water moves into the cell and the cell starts to swell.
Let's talk about osmosis. If we look at its simplest term, in terms of a jar, we have a semi-permeable membrane down the middle. Through that membrane, water can move. Solutes, osmoles, can't. So if we have lots of solutes in this side and only a few in this side, which way is water going to move? Which way is our osmotic gradient? Remember the glass of squash. Water is going to move to dilute. So it's going to move from an area of low osmolarity to an area of high osmolarity. So water is going to move that way. Until such time, as with all gradients, we reach equilibrium. And then we don't have a gradient because we've got equilibrium. So in this case, water will have diluted the osmoles, the solution, and we are now equal. So I've got, what, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6 on either side. At this point, water, although it can still move, it'll move backwards and forwards equally. Yeah? No osmotic gradient. In terms of cells, the plasma membrane, we'll talk about it later, is not permeable to water normally. So in terms of cells, we have to have water channels. So we have things called aquaporins. Aquaporins are water channels. So depending on the gradient, water will move in or out of a cell. It will move from the intracellular compartment to the extracellular, or from the extracellular to the intracellular. Generally, in normal healthy people, we have no gradient in terms of osmosis. So we have equal movement of water. We have something called isotonicity. Whoops. Isotonicity. Yeah? Equal movement of water, equal osmotic grade, no osmotic gradient at all, equal osmolarity. Okay, so let's look at what happens in cells when we change that. If I have a cell, and if I have more solutes within the cell than outside, so we'll just have a couple outside, just for good measure. Which way is water going to move? Yep, it's going to want to dilute, remember. Like our glass. It's going to want to move into the cell from the extracellular compartment to the intracellular compartment. If this happens, what do you think is going to happen to that cell? You put water inside it, it's going to swell. It's going to swell, it's going to swell. Eventually, it could actually burst, but it will certainly start to swell. So it will swell up. This situation where we have the extracellular compartment, we have low osmolarity, if you like, or low tonicity, is called hypotonic. In other words, it's low in the extracellular compartment, water moves into the cell, cells swell. Okay, so let's look at the alternative. If we have a cell, and say, for example, we haven't had a drink, the extracellular compartment gets more and more dehydrated, lots of solutes, very concentrated. In the extracellular compartment, there we go, lots and lots and lots, lots and lots, only a few in there. What's going to happen? The opposite. So water is going to move from outside the cell into the extracellular compartment. As a result, what's going to happen to our cell? Yes, it's going to start shrinking. So in this case, it shrinks down. And this is, if that was hypotonic, that was isotonic, this must be hypertonic. The extracellular compartment has more osmotic particles than the intracellular.
Ionic balance involves chemical disequilibrium. So ionic, we're obviously talking about the balance of ions. Balance, though, doesn't necessarily mean that they are equal. It's just refers to an optimal state or homeostasis. Solutes, remember, contributing to osmolarity can be lots of different things, and they don't have to be a specific chemical. It doesn't matter in terms of osmosis what those chemicals are. So therefore the intracellular compartment can be full of K+, hydrogen phosphate proteins, while the extracellular compartment is full of sodium, Na+, and Cl-, different chemicals but all contributing to the osmolarity of those areas. So they all contribute to osmotic equilibrium, but there is chemical disequilibrium. And this chemical disequilibrium is normal. In other words, the type of ions, often clinically called electrolytes, differs between the intracellular and the extracellular environment or compartments. Water and electrolytes across the course of the day have to balance in terms of input and output. So the amounts gained each day of both has to be equal to the amounts lost. On the following chart, study how the chemical composition varies between the intracellular, these are the beige columns, and the extracellular compartments, the extracellular being the red and the purple columns. Note that the red and the purple columns make up the extracellular compartment, whereas the intracellular are the beige columns. Look at the tallest columns. What ions are they? What solutes are they? And what compartment do they relate to? So in summary, Atoms of an element are electrically neutral because they have an equal number of electrons and protons. The outer shell is known as the valence shell, and electrons in the valence shell prefer to be in pairs. In atoms, if they lose an electron, they become more positive. They become cations, like our Na+ or K+. If they gain an electron, they become more negative. They become anions, like Cl-. Atoms combined in various ways via the electrons in their valence shells to become molecules. So they bind atom to atom via electrons to form collections of atoms called molecules. Also, those molecules can then bind to other molecules to become macromolecules. Within the body, there is osmotic equilibrium, so there is no osmotic gradient between the intracellular and the extracellular compartment. Remember, we called this isotonicity. But there is chemical disequilibrium between the intracellular and the extracellular compartments because the solutes differ in those two compartments. All of this creates a normal balance or homeostasis.